How's it going, you guys? So in this video, I wanted to talk about the Krebs cycle, as well as the thing that precedes the Krebs cycle, which is pyruvate oxidation. And so those two processes happen inside the mitochondrial matrix. So if you think about the mitochondria as having two membranes, it has an outer membrane, and it has this inner kind of superfolded convoluted membrane. This right here is the matrix. So glycolysis, if you recall, glycolysis occurred in the cytosol surrounding the mitochondrion, and it took glucose, a 6-carbon molecule, 1, 2, 3, 4, 5, 6, and it broke it down into 2 3-carbon molecules. And those 3-carbon molecules are pyruvate, or pyruvic acid. For our purpose, pyruvate and pyruvic acid are going to be treated as the same thing. So this pyruvate, once it has been created or generated, this pyruvate will enter the matrix to enter pyruvate oxidation, as well as the Krebs cycle. So that is just a big picture look at the Krebs cycle. So now let's talk about the details. So for starters, let's talk about pyruvate oxidation, because pyruvate by itself, as pyruvate, does not enter the Krebs cycle just yet. It needs to be oxidized and needs to be converted into something else. So this pyruvate, which is a 3-carbon molecule, will go through a series of steps. For one, it will lose one of these carbons as CO2. That's because if you look at a pyruvate molecule, we have our 2 carbons, and that 3rd carbon is actually a carboxyl group without the H, so it's ionized. So this right here, this carboxyl group, will leave as CO2. So if we just lost a carbon, we now are left with a 2-carbon molecule. Now I'm going to erase this so it doesn't get in the way, but I just wanted to go into that detail so you guys can understand it a little bit better. But we lose a carbon as carbon dioxide. We also, NAD+, we also get oxidized. So if you remember, Leo says GR, lose electrons, oxidation, gain electrons, reduction. So pyruvate gets oxidized because it loses electrons to NAD+. So NAD+ will pick up those electrons, and it will become NADH. So it picks up electrons and a hydrogen, and there's also an additional hydrogen ion that is generated here. So at the end of this process, we have a 2-carbon intermediate, and then shortly after, we have this thing called CoA, or coenzyme A. And coenzyme A, with the help of an enzyme, this whole process is enzymatically catalyzed, so every single step that we talk about here uses an enzyme. We just won't talk about those enzymes in any detail. But this coenzyme is attached to this 2-carbon molecule, and we generate this 2-carbon molecule with the coenzyme A, and we call this molecule acetyl-CoA. So that is one molecule that I will require that you know the name of. So you need to know the name of pyruvate, which is what we start with and what we end with in glycolysis, and you will need to know acetyl-CoA. Now, this whole process, we'll consider this whole process pyruvate oxidation. Pyruvate oxidation. So this precedes the Krebs cycle. So now that we have acetyl-CoA, now we can enter the Krebs cycle. So the Krebs cycle starts off with this 4-carbon molecule. And you don't need to know the name of this 4-carbon molecule. There's only one molecule within the Krebs cycle that I will require that you know the name of. But we start off with this 4-carbon molecule. We then pull in acetyl-CoA. And by the way, for pyruvate oxidation, we'll call this step 1, where carbon dioxide leaves. We'll call this step 2, where we remove electrons, we oxidize pyruvate, and we reduce NAD+ to make NADH. And then we'll call this step 3, where CoA gets attached, forming acetyl-CoA. So now I want to start over with the steps, because now we're entering the Krebs cycle. This step is going to be step 1 of the Krebs cycle, where the 2-carbon acetyl-CoA is kind of pulled in to the Krebs cycle. We generate a 6-carbon molecule in the process, because we add these 2 carbons to these 4 carbons, generating a 6-carbon molecule, and CoA is kicked off in the process. If you recall, enzymes cannot be or do not get consumed by a reaction in the same way coenzymes don't get consumed by a reaction. So by the end of this process, we'll have CoA that we can now reuse. We'll recycle that back to step 3 of pyruvate oxidation. But CoA or coenzyme A was necessary because without it, we would not have been able to stick these 2 carbons to this 4-carbon molecule. And this 6-carbon molecule has a name that you will need to know. It is citrate, or citric acid. I'll put citric acid. And that is the main reason why we also call the Krebs cycle the citric acid cycle. So, because we generate citrate in the process. So that was step 1. We kicked off CoA, and now we have a 6-carbon molecule. We already got rid of one of those carbons in the first step of pyruvate oxidation. So now we have 2 more carbons to go. So during step 2, we'll basically do the same thing that we did in step 1 of pyruvate oxidation. We'll pull off another carbon dioxide, another CO2. We'll also, in this same step, take NAD+ and generate NADH in addition to a hydrogen ion. And I'm also going to move the camera over just a little bit so you guys can see this a little better. So we will essentially do the same thing that we did in the first 2 steps of pyruvate oxidation, and that'll kick off a carbon. So now we go from a 6-carbon molecule to a 5-carbon molecule. Now again, you don't need to know the name of this molecule. You won't need to know the names of any of these carbon molecules moving forward. Just know citrate, acetyl-CoA, and pyruvate. So now in step 3, I'm going to make it kind of big because there's a lot that happens during step 3. We pull off another CO2. So this is the last carbon that we need to get rid of. So we got rid of 1 carbon, 2 carbons, and 3 carbons. So now we got rid of all the carbons that were originally in pyruvate. And we also take NAD+, pull off electrons, reduce NAD+ to become NADH, and of course an additional hydrogen ion. But here we also have some substrate level phosphorylation going on. So we pull off a phosphate, or rather we just take the energy, sorry I misspoke there. We just take the energy generated in this step, because this is an energy releasing step, and we use that to take ADP, adenosine diphosphate, 2 phosphates, plus an inorganic phosphate, and we generate ATP, adenosine triphosphate. So we take the energy that was released by this particular step and make 1 ATP. And then we're left with a 4-carbon molecule. Now, these last few steps, I'm not going to require you to know the details of, but we have this 4-carbon molecule. It's chemically different from this 4-carbon molecule, so we have to chemically change some things around in 3 steps. So we have steps 4, 5, and 6. And during this process, we also generate FADH2. We'll take FAD, make FADH2. FADH2 is just another electron carrier, just like NADH. So we have FAD, create FADH2. And then here we take another NAD plus and generate NADH and an additional hydrogen ion. And this whole process right here is the Krebs cycle, or citric acid cycle. Krebs cycle. So those are the details. Pyruvate oxidation comes first, and then the Krebs cycle, and all of this happens in the mitochondrial matrix. So just to kind of summarize things up, I'll move this here so we can see everything. So just to kind of summarize everything, during this whole process between pyruvate oxidation and Krebs cycle, we made 1, 2, 3, 4 NADHs. So 4 NADHs. We made 1 FADH2. So 1 FADH2. We made 1 ATP molecule. 1 ATP. And we also made, as a byproduct, a waste product, 3 carbon dioxide molecules. So 3 CO2s. Now, I will encourage you guys to go back to the beginning of the video, or at least think back to the beginning of the video when we summarized glycolysis. At the end of glycolysis, for every 1 glucose molecule, you get 2 pyruvates. So for every 1 glycolysis, we get 2 pyruvates. So we have to go through this whole process twice. So really, we have to double everything if we want to be accurate. So instead of 4, we had 8 NADHs. Instead of 1, we have 2 FADH2s. Instead of 1 ATP, we have 2. And instead of 3 CO2s, we actually have 6. And this makes sense because 1 glucose molecule is C6H12O6. It has 6 total carbons. So those 6 total carbons end up as carbon dioxide. And these 2 right here, NADH and FADH2, they are both electron carriers. So they are going to head to the next step of cellular respiration, which is the electron transport chain, or the whole thing is oxidative phosphorylation. So I will put ETC, and I will make a separate video on the ETC and oxidative phosphorylation, so you guys can watch that if you would like. So that's the whole process. Hopefully that made some sense. Hopefully this is a good review video for you guys, and I will see you for the next video.